Professor Marignani, thank you so much for joining me today.
Thanks for the opportunity.
Professor, tell me why you chose these two particular chemicals and thought that they might work together well to inhibit cancers.
Well, the current preclinical trial that we've just published is actually based on the previous study. The previous study that was published in PLOS One in 2013 characterized or described the animal model. So we had to come up with the animal model and actually design the animal model. And once we designed the animal model, we selected the cells from the tumors, and we were able to grow these cells in tissue culture in Petri dishes, and we began a series of tests on various chemicals and compounds to see which would best inhibit some of the proteins that we were interested in.
You'll have to explain to a layman what the animal model is.
So the animal model is where we're able to take the DNA of mice, re-engineer it so that it will behave differently, and put the DNA back into the animal, into the mice. And so this particular mouse model actually will develop HER2-positive breast cancer. And then we went in and we removed another gene. And as a result of that, the mice develop very metabolically active and aggressive HER2-positive breast cancer.
Now, when it comes to cancers, there's all kinds of different kinds of cancers, correct?
Yes, there is.
So is breast cancer one kind, or are there even variations in breast cancers?
Well, we know, based on some work that was also done here in Canada and British Columbia, that there's up to 12 or 13 different types of breast cancer, right? And we're learning more and more every day that there might be subgroups within the different types as well. So it's not a straightforward disease. When we think of cancer, it's a very complex disease, which is why some women respond to treatment well, and other women with what is supposed to be a similar cancer don't respond as well because of the complexity of the individual cancers.
Is this HER2 cancer a common one?
It's roughly 15 to 25% of the women with breast cancer would have this type of breast cancer. And even within that population, there might be differences. As I said, some women respond very well to the treatments, and some don't.
What do these two new chemicals do together that they don't do separately, sort of thing?
Well, individually, they both work very well at inhibiting specific protein communication pathways. One of them will inhibit a major protein that's involved in all cancers. And in people without cancer, these proteins, I have to say that these proteins actually function normally in our bodies. It's when the function is lost or there's something wrong with the function, that's when we get the cancer. So the first drug actually inhibits a major, what we refer to as a protein signaling hub. So it's almost like a roundabout. There's about 100 roads coming in and 100 roads coming out. And if everything's working perfectly, the traffic moves smoothly. In this case, if something goes wrong with this major protein, accidents happen, and in this case, cancer happens. The other compound inhibits basically the energy of the cell, of the tumor cells, because cancer cells tend to be very energetic. They produce energy, and that energy that they produce, they then use to continue to grow. And so we've managed to come up with a way to turn the energy off. And so the two compounds, when they're introduced at the same time, they actually have a greater effect. So individually, they both work well to inhibit tumor growth. It's when we combine them that we actually see a better inhibition.
Why has someone not combined these two before then?
I really don't have an answer for that, but the way we designed the mouse model, so in our previous paper, I was trying to find strategies that would target these two major communication pathways. We did, and so we have, and it's here now, so this is good.
Do these chemicals kill the cancer or merely stop it, as it were, so it stays at the same growth level that it is?
No, it actually kills the cancer cells. I mean, in some instances, we could not find tumors in the mice. Because these mice are it's a spontaneous model, so the animals will develop spontaneous tumors, just as humans do. We know that roughly at around 5 months they begin to develop the tumors, and so we start the studies at the 5-month period. So we know that all the mice start off with tumors, but by the end of the study in 21 days, there were some instances where we weren't able to find the tumors, and we knew they had them on day 1.
So this is pretty exciting then.
We're very excited about it, and we're very excited, and I think your question is quite elegant in why haven't these been combined, and so we're very excited, and we've combined them now, and we're continuing the work to try and come up with different combinations.
Professor Marignani, there have been several advances recently in cancer treatments and theories behind it and so on. Is it possible that various aspects of these different procedures can be combined to have a wide-ranging cancer killer kind of thing?
Well, that's a very good question. Again, the pathways, the communication pathways that we are investigating are actually quite common in other cancers and other diseases. And so there is the possibility that this combination may actually work for other cancers. So we do have other animal models of cancer that we're also interrogating with these combinations. So yes, it's true. The combinatorial therapy is probably a good strategy to move forward for the treatment of various diseases, including cancer, because we're coming in from 2 different angles. The majority of the treatments that are currently in clinic today are perhaps attacking one branch of the disease or the communication pathway. By combining compounds or drugs to come in from different angles, I think the clinical outcomes can improve.
These kind of tests, we always hear that they're in the initial stages and it takes years before they could possibly be on the market, so to speak, helping people. I guess this is the case here as well. You still have to go through a lot more trials and so on before it gets to the human stage?
Well, both of these compounds are currently in various phases of clinical trial for other diseases, and so they're already being moved in trials for different cancers and different diseases, but neither of them have been combined. This is the first study that actually combines them, and for breast cancer.
So we could see something like this helping people in the general public relatively sooner than the usual 5 or so years that it might take.
I would say, I'm cautiously optimistic that it would be sooner. The hopes is that we were able to mount a phase 1 clinical trial, because without that, it's in the ether, right? We do need to do a phase 1 trial.
What is the next step then, Professor Marignani?
So, as I said, we're in the process of testing other compounds to attack different communication pathways and these protein pathways. And we've got other cancer models and disease models that we're working with, mouse models that we're working with. And so what's also very interesting is we've taken the tumors from these animals. So we harvested the tumors, and we're now looking at the molecular level. We're looking at the genes and the various proteins and the various metabolites that are in play within the tumors themselves and how they have responded to the different treatments. And so we can reverse engineer the pathways that are involved in silencing the tumors.
Well, Professor Paola Marignani, thank you very much for your time today.
Oh, thank you so much.